A warm welcome to all of you. Today we are discussing cell division for growth, for replacement, for repair, for reproduction. What is necessary? New cells need to be produced. How the new cells to be produced? Only through cell division. Here you can see for the growth of a baby to adult, replacement of old cells by new cells, repair, healing of wounds. Yes, new cells need to be produced, right? For reproduction, production of gametes. What is necessary? New cells should be there. Then for the production of new cells, what is necessary? Cell division is very essential.
There are two kinds of cells present in human beings. They are body cells and sex cells. Body cells consist of 46 chromosomes. That means 23 pairs of chromosomes that we can represent 2n. Both male and female consist of 46 chromosomes. 2n means diploid in nature. All body cells are diploid in nature. Then what are body cells? Body cells means all the cells present in human beings except sex cells, we can say. Right? Yes. What are the body cells then? Nerve cell, blood cell, then glandular cells, muscle cells, epithelial cells. These are examples for body cells. Then sex cells. What are sex cells? Sex cells are produced from sex organs of human beings. That means male sex organs produces sperms, female sex organ produces egg. These sex cells consist of half the number of chromosomes of body cells. That means 23 chromosomes. You can see all sex cells of human beings. Both male and female is sperms and egg. Both have 23 chromosomes. That we can represent N, that is haploid in nature, half the chromosome number of body cell. Then how new baby will form? This sperm fuses with egg to form new baby. Then sperm consists of 23 chromosomes, egg also consists of 23 chromosomes. These sperm and egg fuses to form is new baby. That new baby consists of 46 chromosomes. That means 23 pairs of chromosomes. That we call it as fertilized egg.
Let us learn cell division. There are two kinds of cell division occur in human beings. Mitosis. First one, mitosis, and second one, meiosis. Mitosis is for growth and development. Meiosis for the production of gametes. That means production of sex cells, male sex cells and female sex cells. What is interphase? Interphase is a preparing stage of cell for cell division. This is also known as resting phase. Why we call it as interphase is a resting phase? Because there is no change in chromosome occur. There is no change in chromosome is visible externally. That's why that is called resting phase. But actually that is a preparing stage of cell for the next cell division. Interphase is a preparing stage of cell for next cell division. You know that interphase active in synthesizing DNA. In the last video we learned, right? DNA duplication occur in interphase. That is active in synthesizing DNA. You know that Yes, there are different types of cell division are there, mitosis and meiosis. Mitosis for growth and development, meiosis is for production of sex cells, right? Yes. Then the new cells at the end of mitosis. New cells at the end of mitosis is relatively small. Daughter cells, we can say daughter cells. Daughter cells at the end of mitosis is relatively small. Then interphase prepares these cells, these daughter cells, for the next cell division and grow to the same size as the mother cell. Yes, then we can learn what are the different phases of or different stages of interphase. Interphase of a cell has three phases. First is phase of interphase, that is first growth phase or G1 phase. What are the different changes occurring first growth phase of interphase? Here RNA and protein are synthesized, volume of cytoplasm increased, mitochondria and chloroplast divide. These are the different changes occur in first growth phase or G1 phase of interphase. RNA and proteins are synthesized, volume of cytoplasm increased in that cell, mitochondria and chloroplast divide. Synthesis phase or S phase is the second phase of interphase. What are the changes occur in synthesis phase? First point, active synthesis of DNA. The chromosomes are duplicated. Chromosome duplication, that means active synthesis of DNA duplication occur in synthesis phase. Then third phase of interphase, that is second growth phase. Second growth phase, that is G2 phase. G2 phase is the shorter growth phase compared to other two phases of interphase, which is the shorter phase, that is G2 phase or second growth phase. Here RNA and proteins continue to be synthesized. These are the main changes occur in second growth phase. Then which is the longest phase of interphase? That is very important question for examination. Longest phase of interphase, that is G1 phase. Yes, first growth phase. But second growth phase is the shorter growth phase. Mitosis. Mitosis is one of the important type of cell division occur in organisms, both plants and animals. Yes, what is mitosis? The cell division in which one parent cell divides into two identical daughter cells. One parent cell divides into two identical daughter cells. That is called mitosis. This mitosis we can see in body cells. Body cells divide into two for growth, for repair, for replacement, for asexual reproduction. Then these mitosis completed in two steps, mainly in two steps. What are the two steps involved in mitosis? Karyokinesis and cytokinesis. Mitosis is completed in two steps, karyokinesis and cytokinesis. Karyokinesis is the first step of mitosis. Karyokinesis means division of nucleus. Mainly there are four phases are there in karyokinesis. They are prophase, metaphase, anaphase, and telophase. Both plants and animal cells, mitosis phases are similar. Four stages of karyokinesis of mitosis are prophase, metaphase, anaphase, and telophase. First, we are discussing prophase. What are the identifying features of prophase? If you are observing a cell through the microscope, how do you know that that cell is coming under prophase? Yes. First point, the nuclear membrane and nucleolus disappear. In this picture, you can see there is no nucleolus. Nuclear membrane starts to disappear. Then, the next point, chromosomes have become short and thick and visible inside the nucleus. Chromosomes are short and thick. The next point, each chromosome has two chromatids. That also we can see in this picture. Two chromatids are there in each chromosome. Chromosomes are short and thick. Then the next point, two sister chromatids remain attached at a particular region called centromere. Then the next point, the centrosome splits into two along with simultaneous duplication of centrioles contained in it. Here you can see, yes, these are the two, yes, daughter centrosome. Yes, centrosome splits into two, yes, along with simultaneous duplication of centrioles. Centrioles also is divide. Then that you can see in this prophase stage. Then the next point, the number of fibers that appear between the two daughter centrioles, which are called spindle fibers. Spindle fibers you can see between the two daughter centrioles. These are the identifying features of prophase. Yes, the next stage of karyokinesis, that is metaphase. What are the identifying features of metaphase? First point, the duplicated chromosomes arranged on the equatorial plane. You can see here all the chromosomes arranged on the equatorial plane. That point you have to remember, on the equatorial plane. That is the identifying feature of metaphase. The next point, each chromosome gets attached to spindle by its centromere. Here you can see, yes, each chromosome gets attached to spindle. This is spindle fibers that attach to the spindle by its centromere. These are the identifying features of metaphase. The next stage of karyokinesis, that is anaphase. Yes, we can discuss the identifying features of anaphase. You can observe this diagram. Yes. Then, what is the first point? The centromere attaching the two sister chromatids divide and separate and are drawn apart towards the opposite poles. Here you can see centromere attaching the two sister chromatids divides. Yes, you know that duplicated chromosome has two sister chromatids. These two sister chromatids divide at the time of anaphase. Then that will separate and drawn apart towards the opposite poles. Here you can see, yes, that will drawn apart towards the opposite pole. Then the next point, the drawing action is performed by the contraction of spindle fibers. This how that will draw apart, by the action of spindle fibers, because you know that spindle fibers attached to is the centromere of chromosome. Then at the time of anaphase, what is happening here is these sister chromatids drawn apart.
Here you can see that will divide. Centromere also divide. The centromere attaching the two sister chromatids divide. Then that will separate. These two sister chromatids separate and drawn apart towards the opposite poles. The last stage of karyokinesis of mitosis, that is telophase stage. Yes, what are the identifying features of telophase? First point, each chromatid or daughter chromosome turn into a thinner chromatin network. Here you can see in this picture, yes, chromatid turn into thinner chromatin network. Then the next point, nuclear membrane reappears. Then nucleolus reappears in each daughter nucleus. Then in telophase stage, nuclear membrane. Here you can see this is nuclear membrane starts to appear. Yes, nuclear membrane and nucleolus reappears in each daughter nucleus. Then the next point, in cleavage furrow starts deepening in the animal cell. In animal cell, you can see a cleavage furrow in telophase stage. But in plant cell, you can't see furrow. Furrow formation is not there in plant cell. But instead of furrow formation, in plant cell, cell plate formation you can see. Why there is no furrow? Because you know that in plant cell, there is the presence of cell wall. Yes, cell wall is there. That's why there is no furrow formation in plant cell. But in animal cell, furrow formation you can see. These are the identifying features of telophase. Then one more point, one more thing you have to remember while labeling the diagram of each phase of karyokinesis. Based on the phase, based on the identifying features, you have to label the diagram. So here, you don't label spindle fibers because there is no spindle fibers here. You can label as nuclear membrane, you can label nucleolus, you can label, then chromatin fibers you can label. You don't label here chromatids because there is no chromatids in telophase stage. That will turn into chromatin network or chromatin fibers. That's why you have to label chromatin fibers. Like that, based on the phases, based on the identifying features of each phase, you have to label the diagram of each phase. The last step of mitosis, that is cytokinesis. What is cytokinesis? Division of cytoplasm. Yes, what are the main points you have to remember in cytokinesis? In animal cells, at the end of telophase, a furrow appears in the cell membrane in the middle, which deepens and finally splits the cytoplasm into two. Here you can see is furrow formation that divide cytoplasm into two, thus producing new cells. Is division of cytoplasm occur here. But in the case of plant cell, instead of furrow formation, what is there? A cell plate formation. Then during cytokinesis, the cell plate grows from the center to the periphery. Here you can see from the center to the periphery, resulting in two cells. Here is when it will grow from the center to the periphery, cytoplasm divide into two, then form two daughter cells. What are the differences between mitosis in animal and plant cell? In animal cell, asters are formed, but in plant cell there is no asters. We know that animal cell has centrosome. Centrosome consists of asters and centrioles. This is helping for cell division. But in plant cell, there is no asters. Then, in animal cell, cytokinesis by furrowing of cytoplasm. But in plant cell, cytokinesis by cell plate formation. There is no furrow in plant cell because of the presence of hard cell wall. Third point, mitosis occurs in animal cell in most tissues throughout the body. But in plant cell, it occurs only at the growing tips, only at the meristematic region. Mitosis occur in plant cell. That's why while doing the practicals of mitosis, different stages of mitosis, we will take onion root tips. We know that only at the growing tips is mitosis will occur in plant cell. What are the significance of mitosis? GR square is growth or increase in body size, repair of damaged and wounded tissue, replacement of the old and dead cells. Then first letter we can remember like this, GR square is growth, repair and replacement. Then the another point you have to remember, maintaining the same chromosome number in the daughter cell. Is after mitosis, the daughter cell will maintain the same chromosome number. Suppose in human body, we know that all the body cells consist of 46 chromosomes. After cell division, after mitosis, the daughter cell also consists of 46 chromosomes. Then mitosis maintaining the same chromosome number in daughter cell. The next point, asexual reproduction in which unicellular organisms. Mitosis occur in unicellular organism at the time of reproduction, that is asexual reproduction of lower organism. Which type of division will occur? Mitosis. These are the main significance of mitosis. Then at least 4 points you have to remember. GR square, this is easy, right? GR square, growth, repair and replacement. Then another point, maintaining the same chromosome number in daughter cell. Revision. Differentiate between the following. First one, mitosis in plant cells and animal cells. That we already discussed just now. You can go through that table. Then the next one, karyokinesis and cytokinesis. Karyokinesis means division of nucleus. Cytokinesis means division of cytoplasm. G1 phase and S phase of interphase. G1 phase is, what are the changes occur in G1 phase? Protein and RNA synthesis occur in G1 phase. Then S phase, DNA duplication or DNA synthesis occur in S phase. The next question, metaphase and anaphase of mitosis based on the position of chromosome. Metaphase, in metaphase, chromosomes are arranged at the equatorial plane. But in anaphase, what is the position of chromosome? Two sister chromatids divides and the two sister chromatids of each chromosome separate and are drawn apart towards opposite poles. That is the main differences between metaphase and anaphase. In metaphase, chromosomes are arranged on the equatorial plane, but in anaphase, sister chromatids drawn apart towards opposite poles. Thank you, children. Have a good day. If you like my channel, please like, share and subscribe. Thank you.